Good morning. Good morning, Hannah, and good morning to the folks here in the room. Thank you for participating today if you're coming in from outside the office or you folks that are from the office. My name is Mike Winegar. I work with Qmed Consulting. I do a fair amount of the regulatory and quality type activities. My background is largely medical device, largely vascular medical devices, some combination products, some stem cell products. Have worked with Medtronic, Boston Scientific, EV3, and a variety of startups. First, just a little bit about Qmed. As you are familiar with Qmed, especially you folks that work here, a variety of different services provided by Qmed, the first being consulting services in various areas, clinical, regulatory, and quality. Secondly, commercial healthcare. We have a couple of specialists that work with reimbursement activities as well as in the U.S. as well as in Europe. Subcontractor, monitoring, CRA, CTA type activities. And we are a global alliance. We have people obviously from the U.S. as well as Europe and other countries associated with Qmed, and a preferred partner for a lot of hospital-based clinical trials, especially within Europe. All right, we're going to talk today a little bit about 510Ks, specifically a case study related to draft guidelines and their impacts on the 510K process in the United States. As you know, the 510K process has gone through a lot of changes in the last several years. Some of them have been by legislation. Some of them have been by the introduction of draft guidances. Back in the day, 10 years ago or so, draft guidances were just that. They were draft, and FDA would not require compliance with the draft guidance until it was actually made official and released. The draft was circulated for comment by industry. Industry would comment. FDA would change things sometimes withdraw the draft, sometimes release the draft. Recently, we've seen a different kind of activity where FDA is issuing draft standards and then saying that they are state-of-the-art and recommend compliance with them, in some cases requiring compliance with them. We're going to talk about a case study today related to the biocompatibility guidance that came out as a draft in 2013 that implemented some changes that are now being required by FDA. The old guidance was called G95, went back to it's a 1995 guidance, nothing too magical there. It was very basic from a 10993 standpoint. We'll get into that a little bit later. From the standpoint of what has changed from the G95 in general, information related to colorants and other chemicals. It used to be that manufacturer, Dow or whoever, would have a additive package that would include chemicals and plasticizers and stabilizers that were considered to be company confidential. That was their secret sauce that made their Lexan. What FDA has started to do is say that you need to know what those materials are, and you need to prove that as subcomponents they're not toxic, not just as part of the mix. So a lot more testing related to chemical constituents of materials. Some of these are extraction methods and very strong solvents, things that a device arguably would not see in normal human use. Some of the extraction times have changed. Some of the testing has changed. Back in the old days with pacemakers, you could take a pacemaker can and just take a piece of the can and run that through your biocompatibility test. Now companies are being required to use full devices instead of what was called coupons before. Obviously that changes the cost involved from a sample standpoint in having to run the biocompatibility screen. The example that I want to talk about is a real-life example. living it right now. It's an apheresis system, and been around for 10 years. They changed the polypropylene connector within the system from one polypropylene to another, and these are just little connectors that hook up silicone tubes. And you guys know what silicone tubes are, right? Yeah. And the reason for the change was the materials being phased out. It just wasn't worth it to the manufacturer of the resin to make that resin anymore. It just wasn't enough volume. So it went from one resin to another. No colorants, no additives, just straight polypropylene. You look at an FTIR, they were indistinguishable from another. The company had a master file on site with FDA, although it was in compliance with USP requirements for Class 6, not necessarily 10993 requirements. We went to FDA, said, hey, the company's changing this out, and we want to, reference their material, and we ran a cytotoxicity screen. Everything was fine. FDA came back and said, hold up there. We need you to do full screen on this. Now, one thing that was good is we just had to do it on the material, not the entire finished device. In our case, it would have actually been cheaper to run it on the finished device because of the surface area and the weight you need. We ended up having to have a special run of 1,000 connectors made just to do biocompatibility testing. So in summary, we were required to comply with the draft guidance. The original biocompatibility screen for the standard, the things that were currently approved by FDA as guidance was $23,000. The additional 3 tests we had to run added $35,000 to the biocompatibility screen. This is just a quick summary of the tests that used to be required for a externally communicating device, blood path and direct. So this is the tubing sets that are used with the apheresis systems. There's several categories of tests here. The cytotoxicity, the sensitization, all of these are also standard ISO EN 10993 tests in the different categories. The 2 categories that we ended up having to do additional tests, and it's not just us, this is very common. A lot of companies are running into this. We're in the genotoxicity and the hemocompatibility areas. This next slide shows the additional tests that were required. The genotoxicity, normally if you do an Ames genotox test, it's a bacterial mutagen test. You apply an extract, 2 extracts actually, one from a polar, one from a non-polar material, and if your bacteria don't mutate and they're tested for mutation as to their antibiotic resistance, then it's considered to be non-genotoxic. These additional tests are much more involved and involve mammalians, mouse testing for blood micronucleus and lymphoma, and very expensive, time-consuming tests to run. Whether there's value added with this is hard to say, but FDA is requiring all 3 of these now for a standard genotox screen. From a hemocompatibility standpoint, before it was very much hemolysis and platelet aggregation tests that were required. The complement activation, that's a C3A complement activation, is a relatively simple test. The next one is the complicated one, dog thrombogenicity. This is actually taking pieces of material and doing dog jugular implants, venous implants, one on the left, one on the right, treatment on one side, control on the other. And it's a comparative test because there is actually no pass-fail criteria. You take the material out and look for thrombus formation on the material itself, as well as irritation to the vein intimal lining. The problem is, if you're making a connector and it's for hooking up 1/4-inch tubing, you're not going to get that in any dog jugular. You're going to have to take a sliver of that material, by nature causing a potential reaction from having an edge that can either be a place for thrombus to begin to form, or cause irritation to the lining of the vein. So plus two, from an animal rights standpoint, you're killing a dog to do this test, and there's a lot of negative public opinion related to that. These additional tests, besides the $35,000 in cost, resulted obviously in additional time, another 6 weeks of time or so added to the testing regimen. From an overall standpoint, it's not just industry that is questioning FDA's motives on this. This is an excerpt from a letter written to Commissioner Hamburg, who, I don't know if you've heard, she's also phasing out of her role at FDA, by a Senate subcommittee asking FDA, what's with implementing draft standards? Legislation requires that these be release standards before they're required to be complied with. And again, this is relatively recent. It's from May. As I mentioned, Commissioner Hamburg's on taking a different position. Christy Foreman, who was head of CDRH, has already transitioned out and is now with the tobaccos group at FDA. So there is going to be a change in the top management at FDA, so to speak. We've seen a congressional shift as well. The Senate is now Republican controlled. These things may impact the device industry. They may impact some of these implementation of standards because, at least in the U.S., Republican controlled houses of Congress tend to be more industry friendly and therefore less regulated from a device standpoint, but we'll just have to see. That is the end of my formal presentation.